So we're going to continue in Chapter 7. As I mentioned last week a couple of times, Chapter 7 and 7 and 8 really go cohesive together, but they've been separated for whatever reason. So additionally, a lot of this information we kind of crash course in lab a couple of weeks ago when we did the first nerve exercise. And so we're just repeating some terminology. We have a basic appreciation that a nerve cell establishes a resting membrane potential by use of the sodium-potassium pump to create gradient for each sodium and potassium. The role of the leaky channels have a factor in that resting membrane potential. And so once that resting membrane potential is established, a cell can use that potential to do some type of work, such as generating an action potential. But to understand that, we've got to be familiar with these terms, including not only just resting membrane potential, but what else? If another term that we could define is depolarization. As you guys discuss this with your classmates, how can we describe depolarization? The membrane potential becomes less negative. Very good. When compared to the resting membrane potential. Which ion did we say contributes to depolarization? Sodium moving into the cell. Sodium moving into the cell. So if the sodium moves into a cell, if it's at rest, sodium moves in through some type of chemically gated, mechanically gated, whichever gated channel, the cell will depolarize. Conversely, a cell may also repolarize. What happens during a repolarization? Very good. Potassium moves out of the cell. So after a phase of depolarization, if gated potassium channels open up, the cell will repolarize. Trying to get back to what? Trying to go back to resting membrane potential. Additionally, nerve cells may actually undershoot resting membrane potential. What is that caused or called? Hyperpolarization. Very good. If a cell hyperpolarizes, it becomes even more negative than it was at rest. And which ion can contribute to hyperpolarization? Potassium flowing out of the cell. Because the gates are slow. The gates are slow. The ions are fluxing still pretty good, but the gates are usually open. We said there's another ion that could actually contribute to hyperpolarization. Do you remember which one that is? Not calcium. Calcium has a positive charge. It's chloride ion, Cl negative. If the cell's at rest and we open up gated chloride channels, chloride would move which way? Into the cell, dropping the polarity, making the nerve cell even more polar, causing it to hyperpolarize. We wouldn't see that during an action potential, but we will see it in something that we're about to address called a graded potential. Some graded potentials can hyperpolarization do this. What questions do you have about some of the terminology? Depolarization, repolarization, hyperpolarization.
Can you restate what you said about depolarization?
Depolarization is less negative when compared to rest. So if the cell's at rest at minus 70 millivolts, it may depolarize to minus 68. You would say it has depolarized. It's no longer at rest. Right, sodium is floated into the cell. It's depolarized. Additional questions or clarifications? So these terms are applicable not only to all the nerves that we're going to study in this unit, but also in all the muscles that we're going to study. Our skeletal muscles can depolarize, repolarize, hyperpolarize. Our cardiac muscle, depolarize, repolarize, etc. And the concepts are the same. And so as I was mentioning, those terms definitely we're going to use during an action potential, but not every stimulus which a nerve cell receives is necessarily excitatory. But we can still use the word depolarization, repolarization, or even just hyperpolarization. So we studied an action potential or a nerve impulse, as illustrated in the graph on the far right. However, I want to bring into play these things called graded potentials. Graded potentials are still potential changes away from what? Resting membrane potential. So a cell may have a graded potential change, and some stimulus, chemical, mechanical, thermal, electrical, whatever, is present. It causes the cell, the post-synaptic cell to do what? To either, this first one represents depolarize, or maybe even hyperpolarize. But there's some stimulus is represented by the little pink arrows. If the cell receives a graded, or I guess generates a graded depolarization, is it going to generate an action potential magically? Spontaneously? No. Why not? Because the cell hasn't met its threshold voltage. So just because the cell depolarized doesn't imply that it has met its threshold voltage. Only if threshold voltage is met will the cell generate an action potential. Can you see that visual difference between the two? So the question is, well, if a graded potential doesn't generate an action potential, why do we even study them? Why do they exist? Does anybody know why these things exist? Why might there be a graded potential? It's not an action potential. It doesn't cross threshold. What might be? It causes a change. It'd be a sub-threshold stimulus, and so it indicates a change. So it's still sensory detection. But, yeah, it'd be like that sub-threshold where you're shocking your nerves in the experiment at 0.05, whatever, 0.5 millivolts. There's a stimulus, but just not enough of the stimulus for the cell to react to respond to, by generating an action potential. What we'll find is these graded potentials can be summed together. What does that mean? Added together. If I add multiple graded potentials, a cell might actually get closer to threshold and maybe even generate an action potential. So standalone, the graded potentials look useless almost. Unless we add, well, they can be summed together. In just a couple of minutes, we'll look at the different types of graded potentials and summation. Again, our graded potentials may be either depolarizations, bringing the potential closer to threshold, or they may be hyperpolarizations, bringing the post-synaptic cell further away from the threshold. These graded potentials may happen at any of the sensing areas of the cell. So definitely you can see a graded potential where? What are the sensing parts of the nerve cell? We have where would those receptors be located? The membrane of the dendrites, membrane of the hillock, and even between there would be the cell body. So the dendrites, the cell body, and the hillock is where you're going to see any potential graded potentials. The axon itself is going to have the ability to generate action potentials. So that would be exclusive over to the axon and the axon terminals. So graded potentials are sometimes described as being dissipating. So if a weak stimulus is present and goes away, the cells, those gated channels are going to close, and the cell's going to go right back to resting membrane potential. Get ready to receive a new stimulus. The action potential is often referred to as an all or none event, meaning if we reach threshold, all of this is happening. get a depolarized, flip the polarity, either positively charged, and then repolarize, and then hyperpolarize, and then quickly establish back to resting membrane potential. What questions do you have at this point differentiating a graded versus an actual potential? So the key is graded potentials are deviations in potential which are where? Below threshold. So you would say subthreshold changes in potentials. They may be depolarizations, they may be hyperpolarizations. If we were to give them a proper name, you can refer to them as an excitatory post-synaptic potential. So you may call them EPSPs, excitatory post-synaptic potentials. Before we look at this in much detail, let's look at the word, let's look at this phrase backwards. So we're talking about what? Potential changes at a synapse. Which cell at a synapse? The receiving cell. So these are potential changes in the target cell, basically, at a synapse. And that target cell is excited in the presence of some neurotransmitter. When we say a nerve cell is excited, we basically say it does what? Depolarizes. And so an excitatory postsynaptic potential is just a graded depolarization in the target nerve cell. You think about the billions and trillions of nerves in your body. One is the source, the other is the target. So if the target nerve depolarizes, we can say it's undergoing an excitatory postsynaptic potential. How many excitatory postsynaptic potentials are shown in this image? 2. Actually, there's 3 grades shown, but just 2 of them are excitatory. So we see 2, 1, 2 that are depolarizations. So these 2 would be the excitatory postsynaptic potentials. So the target nerve cell receives some chemical stimulus, opens up which gates? Chemical gated channel, so we open for sodium. And sodium would cause the cell to depolarize a little bit. Or we could say the cell's just undergone an excitatory postsynaptic potential. So it's a graded depolarization. Alternatively, what do you think we could call a graded hyperpolarization? Inhibitory postsynaptic potentials. You can abbreviate these IPSPs, inhibitory postsynaptic potentials. And these are graded what? Hyperpolarizations. How many are shown in this image? 1. Shown in this image, a graded hyperpolarization. Why might a cell need to hyperpolarize after rest? Does it rest? Why might it need to hyperpolarize? Why would a cell need to be? Why would you want to inhibit a nerve cell? So to prevent a reaction. So for example, when I'm standing here, I want to purposely stand here. I don't want to start convulsing on the floor, right? But all my muscles could let me convulse on the floor, right? But I need to inhibit muscles so that I don't just start randomly convulsing, right? Additionally, you might want to hyperpolarize some nerve cells if you go for some elective surgery, right? Your surgeon wants to cut on your spine, you're going to wait a minute. Let's hyperpolarize my sensory nerves first. Or they will give you a block. Or something like that. So there's certainly a role in needing to hyperpolarize or inhibit some nerve cells. Quite often, actually. So as mentioned, these postsynaptic potentials can be summed together. Our graded potentials can sum together. What does that mean? So that means they can add together. This is a nice image looking at a single postsynaptic cell. You can see its axon zoomed in on its cell body and its dendrite. We would call that the postsynaptic cell because I can see the axon terminals of other presynaptic inputs. This helps quantitate that in your brain and your spinal cord, it's not just a one nerve to one nerve communication. It may be one nerve to 20 nerves communication and vice versa. And so if you think about the multitude of cells that may be talking, targeting a single postsynaptic cell at the same time, it might be quite numerous. And not all cells, nerve presynaptic inputs cause excitation. Some may also cause what? Inhibition. Okay. And so the axon terminals of presynaptic cells that cause a postsynaptic cell to depolarize, or just simplify by showing the green axon terminals, and the hyperpolarization inhibitory postsynaptic potentials are shown by the red little terminals. The question is, well, if we've got all these inputs, what is the net effect on the postsynaptic cell? And so we've got to look at what's going on. Are all the presynaptic releasing their neurotransmitters at the same time, their chemicals, or are they releasing it in a delayed fashion, right? So what's the effect? How can we sum these together? And there are two types of summation. The first type is called temporal summation. I've got a little brief animation. If you look at, for example, this single presynaptic cell, you can see how its terminals branch out, the axon branches, and then I've got some little arrows. Those arrows are going to flash to illustrate that this cell, this presynaptic cell, is going to release neurotransmitter fast. It's going to release it. Postsynaptic cell is going to depolarize. Before the cell gets back to rest, it's going to receive more excitatory neurotransmitters. So it's going to continue to excitatory, which is going to continue to depolarize. If I release it again, it's going to continue to depolarize and probably get to what? Threshold and generate an action control. So a temporal summation, the key is that a single presynaptic input releases neurotransmitter rapidly before the postsynaptic cell regains resting membrane potential. So it just continues to kind of stair step up to its threshold. So temporal summation, how many presynaptic inputs? One presynaptic cell releasing a neurotransmitter that would be considered to be what? I mean, what type of effect on the target? Excitatory. So one presynaptic cell releasing excitatory neurotransmitter how? Rapidly. So that our depolarizations sum, they add together so that the cell actually reaches, the postsynaptic cell reaches its threshold to generate an action signal. The qualifier here is that a single presynaptic neurotransmitters or nerve cells release the neurotransmitter rapidly. The cell will depolarize, the postsynaptic cell will depolarize, won't quite get to rest. They'll depolarize again, won't quite get back to rest, and depolarize again, and probably escalate up to threshold. Or sum up to threshold. So our graded potentials can be summed by temporal summation. A single excitatory input grades the postsynaptic cell up to its threshold. Does that make a little bit of sense? We don't always have just one presynaptic cell acting at the same time, though. You may have multiple presynaptic cells functioning together, and in that case, you would take it together spatial summation. Okay, so we're looking at the graded potential over an area. So this is going to call into more than one presynaptic input. And so I've got, I think, 1, 2, 3 different nerve cells picked out for this animation. And so in this case, I see one cell green, so it's going to cause depolarization where those terminals are. You can see another one also green, but this one is inhibitory. So I've got 1, 2, 3, 4, 5, 6 axon terminals where an area would cause depolarization, and then 2 that would cause what? Hyperpolarization. The question is, well, what happens? I've got all these depolarizations and some hyperpolarization. What do you think the net effect is? We may depolarize, right? So the inhibitory may offset a little bit, but we still depolarize. Or maybe if they're balanced, what might happen? It may have no net effect, where the postsynaptic cell doesn't even undergo a change. Even though one cell is telling it to depolarize, the other cell is telling it to hyperpolarize, they may actually cancel each other's effects out. They may. Or we may sum up to threshold to generate an action potential. So what's the difference between temporal versus spatial summation? It may, but how could we? They could all be excitatory. They could all be inhibitory. It may be a mixture.
There's more than one presynaptic cell.
There's more than one presynaptic cell. Very good. In spatial summation.
So like for temporal summation, like the neurotransmitters of the presynaptic releases, are they always excited?
Not always. The example we gave was excitatory to bring the cell up to threshold. You could temporally just hyperpolarize and trigger an action potential as well. So is there a purpose to the graded potentials? There is. They can be summed by either temporal or spatial summation.
The spatial doesn't happen rapidly, like.
It may, but the key is that the release of these 3 cells neurotransmitters happening almost simultaneous. And so whether there's another release right behind it, I don't want to say is irrelevant, but doesn't define this. It's just that they're happening nearly almost simultaneous in the same span of a millisecond or so. Good question. Additional questions or clarification? Yes.
And the inhibitory postsynaptic, is that like, would that be happening when your arm falls asleep or something? Like when someone's given an epidural? What's happening?
Kinda. When you sleep, when you lay on a limb, basically you inhibit the blood flow, and then so you basically starve those organs, and so it's going to take a while to get the blood flow back there. So you can also suppress your nerves in that fashion. But if you go for surgery for like a spinal block or something like that, they may cause, I don't know all the different chemicals that they may use, but they could cause hyperpolarization, like opening up chloride ion channels, or even just blocking sodium channels. Why would you want to block sodium channels? Yeah, if you block sodium channels, the cell would never depolarize to threshold. And so that's kind of a really easy way to do it. Just temporarily block up the sodium channels to prevent depolarization. Doctor can cut away. Threshold not met. No problem. So that stuff wears off. Good questions. So I want you to test yourself. I want you to plot and label an action potential. Include, although your book uses phase 1, phase 2, I want you to use depolarization, repolarization, hyperpolarization. And then mark the threshold, resting membrane potential, and then after labeling the phases, identify which ions in which direction they would be flowing across the nerve cell membrane. So test yourself for me. All right, so we went through and plotted an action potential. We can check your work. This is a single action potential. We can see the threshold represented at about minus 55 millivolts, the resting membrane potential at about minus 70 millivolts. When some stimulus is received, the cell will, we can now call this what it would depolarize or grade, it would grade up to threshold, and it is a depolarization. So this upward swing would be labeled as depolarization. But what I want to clarify, after doing our experiment in lab a couple weeks ago, we grade up to threshold through some chemical, mechanical, thermal, electrical stimulus. We'll grade the cell up to its threshold. Then we open up what?
Repolarization.
We open up,
Oh, sodium channel.
what kind? What can we open now that we voltage gated sodium channels to continue the depolarization?
Is the peak, like, would you consider the peak the action potential reach?
The whole thing, this from the grading up and then beyond is collectively the action potential. And to be honest, I don't know if there's a proper word, but I just call it the peak of the action potential. If there's a proper word, I don't know what it is. But the action potential is this whole experience. It's, yes.
I think we talked about it, but is it completely different? Because mine is way more held like. Does that mean pointing? Is that important?
No.
Okay, that's not even.
And even in a real, if you're looking at this, if you become a neurosurgeon or whatever, these are gonna probably, depending on this time span, because this is stretching out milliseconds. Your alphas are gonna squish to seconds, it's gonna look like a straight up and down line. And so we're just pulling it out so we can stepwise what's happening inside of the cell. As long as you realize, the biggest thing critical to me is depolarizing to threshold and then becoming positive. And then depolarization, with the angle of the whatever. So the cell will depolarize due to which ion? Sodium. Sodium flowing into the cell during depolarization. The voltage gap. Voltage gated channels will, sodium channels will close when we open up. And what kind of potassium channels they are? Also voltage gated potassium channels open to allow for the phase of repolarization. Potassium is flowing
Out of
the cell. We said those voltage gated potassium channels are slow, so even though we've met rest, the cell will continue to flux out potassium to contribute to a phase of hyperpolarization. And then all the gated channels will close, and the leaky channels are leaking. The leaky channels are leaking. The pump is pumping, so the cell would, that membrane would go right back to resting in the grade potential.
So in hyperpolarization, was that when there could also be polarization?
Yeah, let me clarify that. In an action potential, it's exclusively potassium contributing to hyperpolarization. The chloride ion may contribute to a graded hyperpolarization. So during an action potential, when the cell hyperpolarizes, it's exclusively potassium still flowing out. If a cell's at rest and then hyperpolarizes, it's inhibitory, that could be chloride ion, but that's only in a graded potential. So the hyperpolarization of an action potential is due to potassium only moving out of the cell. Thank you for bringing that up. Correct. So if a cell has a graded inhibitory postsynaptic potential, it could be potassium flowing out, or it could be chloride moving in. But that wouldn't happen.
So that's only on the graded.
On the graded, correct. All right, what additional questions do you have about the phases of an action potential? It took us a couple weeks to start making this comfortable, and we're still working on it on the handout that you're going to submit this week. But we got to add more detail to it. We're going to see what else is involved in an action potential. If you turn to figure 7.20a, it discusses these things called refractory periods. Refractory periods that this term, refractory period, is not exclusive to the nervous system. You can use this term in the cardiovascular system. You could use it in the reproductive system. Okay, so here in the action potential, the refractory period is describing the function of the use of these gates to ensure that once an action potential is started, that it finishes. Okay, so we use these things called refractory periods. There's an absolute refractory period and a relative refractory period is kind of subdivided here by these time phases. During the absolute refractory period, during our depolarization and the bulk of the repolarization, the cell is depolarizing, it's getting, right, it's approaching and then exceeding threshold, right? And then we open up the voltage-gated potassium channels, and we said potassium's flowing out. As potassium's flowing out, what's the probability, so we're taking all this positive charge out, what's the probability of the cell crossing threshold to depolarize? If I'm taking positive out, what's the probability of positive leaking in and causing the cell to depolarize to threshold? It's actually not going to be very likely. All the voltage-gated potassium channels are open and potassium is just rushing right out of here, man. And so even though leaky sodium's coming in, it's not enough to counteract that big efflux of potassium. And so what happens is it ensures that the cell gets closer to rest. Because here's another way to look at it. We don't want our action potentials to sum together, like we did our graded potentials. If our action potentials sum together and we plateau, what can the cell not do? You can't repolarize, it can't rest. If it's not at rest, you can't receive a new stimulus. So it's so imperative, so important that once that depolarization beyond threshold is taken care of, it's critical that it immediately goes right back. Repolarizes, try to get back to rest. And actually we do need that hyperpolarization. Because once it gets into the relative refractory period, it's hyperpolarized. The probability of it crossing threshold now is even less. And so we close those gated channels and the cell will definitely go back to resting membrane diffusion. So these refractory periods are identified to make sure that this all or none phenomenon happens and the cell completes the action potential but gets back to resting membrane diffusion, where it can receive a new stimulus. All right, so the absolute was listed on the left-hand side and the relative during that hyperpolarization. That is an action potential. Here's the interesting, maybe confusing part about it, is those events that's taken us a couple of weeks to talk about happen within a couple of milliseconds within a tiny microscopic part of the nerve cell. The membrane right next to it has to do the exact same thing. And the membrane right next to it has to do the exact same thing. And the membrane next to it has to do the exact same thing. Until we conduct these action potentials towards what part of the nerve cell? Where do we want to get these action potentials to? The terminal, right, to the axon terminal. And so we have to look at how the action potentials are conducted along the action potential, or along the axon, excuse me. So my abbreviation to AP is an action potential. We know that not all action potentials are conducted or carried across the cell membrane at the same rate. It depends on the size of the axon. We did some visual comparisons last week in lab. We looked at, say, the olfactory nerve versus, say, the accessory nerves on the brainstem. Remember, some big nerves, little nerves. We learned what about axon diameter? Conduction is increased when larger nerves conduct faster. Okay, so just looking at diameter, we learned that larger nerves conduct faster. Additionally, we learned that some nerves are myelinated and some are not. We have both cell types in our body. Those cells that lack any myelin Your book calls it conduction along cells without myelin, I think it's all that. Or they could have said contiguous, not continuous, but contiguous. Contiguous conduction is how an action potential is conducted, carried, along a non-myelinated nerve cell, or along gray matter. Conversely, those cells which are myelinated, they conduct by what's called saltatory conduction.
I'm sorry, if the axon diameter is larger than
The rate of conduction, the rate is faster. The direct correlation. That was covered in lab 3, I think it was 3 maybe. The nerve lab.
I have a question about the saltatory conduction. It said in the video that it jumps. Is that, and they said it real fast, so I didn't know.
Yeah, a lot of people describe it as jumping, and I'm going to show an image to hopefully clarify that for you in just a second. So I'll give you an image. I may have it in your guided notes, I'm not sure. But it's described as jumping. But what's important to realize is these are events that are happening inside of a nerve cell. And so
So it doesn't jump, oh, it goes through the myelin, but it just looks like it's flashing.
it goes through the cytosol of the nerve cell. And so let me show you some images, and that might help a little bit.
Okay.
And then I do have a brief animation we can show. So the terminology, contiguous are cells which lack any myelin. Saltatory is how the action potential is conducted. And so instead of saying jumping, I would say conducted along a myelinated nerve cell. And I'll give you some images to help with this. In contiguous conduction, a lot of times the textbooks will describe this as analogous to doing the wave at a sporting event. When you stand up, that would be analogous to depolarizing. And when you sit down, be analogous to repolarizing. And then so one section, say section 301, can depolarize. 302 is in the middle of what? Rest. And then 301 will repolarize. That excites 302, so 302 will begin to depolarize. Well, the next section is depolarizing. The previous section is in the middle of hyperpolarization and maybe even rest. And then so 302 will begin to repolarize. And then section 303 would depolarize. 301 is definitely at what? Rest. Okay. 302 is hyperpolarizing, 303 is depolarizing, and it just continues. And so we don't have people like a wave. We don't have people in your cell membranes. However, we do have voltage gated channels. You can see just open and close. Open sodium channel, voltage gated sodium channels, close them, open voltage gated potassium channels. In the next section, open up voltage gated sodium channels, close them, open up voltage gated potassium, and it just conducts kind of like that along the nerve cell. The daisy chain, the
idea that, you start one reaction, and you'll just keep on moving down.
and it
It's not,
and it'll continue until we run out of the nerve cell, basically. We get to the axon terminal. And then a whole other series of events happen. We'll cover in chapter 8. But contiguous, illustrating that we conduct it from the hillock down the axon towards the terminal and not backwards because of the refractory periods. And those refractory periods make sure that the preceding membrane is hyperpolarized, and so we can't excite it. And it just conducts towards the terminal. If we contrast that to saltatory conduction, this image might help a little bit. It's still intracellular electrical change. It's full and open voltage gated channels. In myelinated cells, though, only segments called nodes of Ranvier of the nerve cell membrane are exposed. And so at those nodes is where you would find the Thom-Salihi channels, but most important, the voltage gated sodium and the voltage gated potassium channels. So when this nerve cell receives an excitatory stimulus, say over here at the dendrites, it'll grade up to its, we can grade up to a cell threshold. And probably, it could be over the cell body, but probably at the hillock we open up the voltage gated sodium channels and then start this cascade. Sodium would flow in at the hillock, and it's inside the cell now, so it just diffuses, right? And it might diffuse back towards the cell dividing, but it's hopefully gonna diffuse as well down the length of the axon. And when sodium ions flow into where this next node is, they voltage change, right? The ions pull up in our voltage gated channel. So sodium would flood into this node. More sodium would flood out, which would diffuse, but they wouldn't affect the gates at the hillock because the hillock is probably in the middle of what? Repolarization and probably even hyperpolarization or rest. And so we can only affect gates at the next section of the membrane. So ions flow in, they diffuse. This section of the membrane would be hyperpolarized. This one's the middle of depolarization. But we flow in and pull the gates in the next segment or the next node right here. So think of it as an intracellular event, not an over the edge hopping kind of thing. Does that help a little bit?
So they don't really jump over the myelinated.
They don't, there's no ions having fun. It's like spaceman, it's not like that. It's just ions flowing inside of the cell. Because our ions are charged, it pulls open those voltage gates and lets more sodium in.
Okay, it's like, but it can only affect like the gates of the next segment. So
Correct.
It can't.
It can only affect the gates of the next segment because the previous segment is in its refractory radius.
Is it only?
Correct. It can only go one way. From the hillock towards the terminal. Does that help at all? Saltatory conduction. We still have the same principles, but it speeds up the rate of conduction so that we don't have to do all these gate openings across the entire length of the cell, only at the what? At the nodes. Then it will end up at the terminal. Figure 7.21 in your textbook tries to illustrate this as well. It shows a pretty good job showing the intracellular chain. And this one's going to the right of the image. This is called saltatory conduction. Let me show you a video. It may or may not help, but let's find it real quick. In this animation, we'll see initially the action potential originated at the hillock. It'll look like a yellow flash, but we don't have flashing. It's just the ions fluxing across a cell. This cell is not myelinated, but we can still see the conduction. All right, so something has caused that cell to threshold up, grade up to its threshold, and generate an action potential. If we were to kind of zoom in, we know that we've got to utilize these gated channels. The pump is working in the background, creating that gradient. We know that sodium is in greatest concentration in the extracellular fluid and potassium in the greatest concentration in the intracellular fluid, as represented by sodium in the purple and the potassium in the blue. This next bit of the animation is just illustrating. So the gradients for each sodium and potassium. We know that through a gated channel, sodium would flow in and potassium would flow out. We've already kind of walked through the phases of an action potential. And here. Okay, so I like this animation because you can actually quantitate the ions fluxing inside of the cell and then how they can affect the permeability of adjacent voltage-gated sodium channels. So I just want you to look at the sheer quantification of the ions. And so when an action potential conducts, opens up voltage-gated sodium channels. So this little section of the membrane where we see sodium fluxing in is doing what? Depolarizing. And then the next section of the membrane will begin to depolarize, while that previous section will repolarize and also hyperpolarize. So you'll see that potassium flowing out in the previous section. Does that help a little bit? And so it's the influx of those sodium ions that pulls the downstream, if you will, voltage-gated sodium channels to conduct that action potential along the length of the nerve cell. And this We know about the pump is pumping, the leaky channels are leaking. And if you're to quantitate it, an action potential happens along each microsegment of a cell membrane. And the question was, well, what's the angle of my peak of my action potential? I mentioned if you were to go to a doctor, they may shorten to just a second, and your action potential is just straight up and down line. Watch that action potential conduct right down the length of the axon. I hope that helped a little bit. Yes or no? And I have an alternative image to illustrate for you cell story conduction, a more structurally detailed image of the myelin and the nodes compartmentalizing where our gated channels are at the nodes. And then the myelin, remember that's a whole other cell that axon associates with the nerve cell. It's actually anchored down. We talk about membrane proteins. We got to anchor that myelin to the membrane to electrically insulate it. That looks like that concludes chapter 7. Let's go ahead and transition to chapter 8. We've got about 15 minutes, so let me pause this. All right, so chapter 8 kind of finishes up the rest of the story. Well, what happens after an action potential is conducted to the end? And there's a whole other series of events at the end of a nerve cell. We haven't even talked about the release of neurotransmitter, and so that's what we're going to address here in chapter 8. How the neurotransmitters release and various types of neurotransmitters, which we'll use this semester. So we're now looking at the end of the nerve cell, looking at the synapse. The title of the chapter is called Synaptic Transmission and Integration. And so what we're going to do is continue that story, talking about what causes the release of our neurotransmitter into a synapse. And we're going to talk about different receptors, specific receptors and how they're built. And then the effect inside of the postsynaptic cell, we have an idea whether they're going to cause depolarization or hyperpolarization. So we'll look at some specific neurotransmitters. We're going to reiterate the role of an inhibitory synapse or an excitatory synapse. But honestly, we really kind of covered this when we introduced the gradient potentials, excitatory or inhibitory postsynaptic potentials. And temporal spatial summation, that is in the wrong chapter, isn't it? We addressed that, and we moved that objective to chapter 7. Role of temporal and spatial summation. And then we'll look at specific neurotransmitters. We're going to look at cholinergic neurotransmitters, amines, amino acids, purines, and neuropeptides. All right. So if we go into the content, to kind of put it in perspective, so far we have been looking at a cell, and we've been kind of honing in on the dendrite, cell body, axon, hillock, and axon itself. Now we've got to transition to the end of the nerve cell and see how we can release this source cell, get it to release its neurotransmitter. And so we've got to broaden our horizon a little bit. So we're still looking at what we introduced in chapter 5 about intercellular communication, how this cell is going to talk to another cell, and we'll call the postsynaptic cell. So here we are. We have 4 nerve cells represented here. Which or how many cells could be considered a presynaptic cell in this image? I would say in this image at least 1, 2, and 3. And this one I'm not sure about. I really don't know about that at the end of that very axon terminal. Your book says it's true, but I don't know for sure. But we'll go ahead and just use it. So this would be each of these would be a presynaptic cell. So where would my postsynaptic cell be? Little guy, the remaining one. So my presynaptic cells would be there, and then the postsynaptic cell, shown in the lower left-hand corner. When this talks to another cell, it would then be the presynaptic, and the other one not shown would be the postsynaptic. So to use similar terminology to chapter 5, our presynaptic cells would be the what? Going back to chapter 5 terminology, the presynaptic cell would be the source, and the postsynaptic cell would be the target. The messenger would be a neurotransmitter. You're going to see neurotransmitter diffusing across clefts from the presynaptic to the postsynaptic cell. Our presynaptic cells may target the postsynaptic cell at its dendrites, at its cell body. I want this thing to go over here to the hillock, or at its hillock. It does have specific names. When the presynaptic cell targets the postsynaptic cell at the dendrites, it's called axodendritic. The synapse is called axodendritic. What do you think we call the synapse where it synapses on the cell body? Very good. Axosomatic. Very good, y'all. Getting your terminology. Axosomatic synapse. And when the presynaptic cell synapses at the hillock, we would call that, that one doesn't give itself away. It's called axo-axonic. So somewhere on the axon. Axo-axonic. So wherever these synapses are, the postsynaptic cell is going to express what? A postsynaptic cell better have receptors. At the dendrites, at the cell body, and at the hillock for specific neurotransmitters. What questions do you have about these various synapses where they may occur at the postsynaptic cell? Axodendritic at the dendrites, axosomatic at the cell body, or axo-axonic at the. It's showing at the terminal. I don't like that, so we're going to call it at the hillock. It definitely can happen at the hillock because that's where our threshold is established. And so if we zoom in at a given axon terminal, okay, we can kind of review that image that we saw in chapter 5. So label the source. Which cell would be the source cell? The top one would be our source. And we could say source, or we could say the presynaptic cell. And the other yellow thing would be the postsynaptic cell. We can see its receptor. And we can also see the presynaptic cell releases neurotransmitters. So the question is, how the heck does the presynaptic cell release the neurotransmitter? First thing is, the presynaptic cell has to do what? Has to depolarize to threshold and generate an action. It has to generate an action potential. However, it gets to the end by saltatory contiguous conduction, an action potential is conducted down the length of that presynaptic cell. So we're conducting an action potential. That changes the voltage, and so we open up what? Do I know what this thing is representing? Calcium channels. They'll be used in conduction, but specialized, we need to hone in at the terminal would be the calcium channels. And so the action potential opens up voltage-gated calcium channels, and calcium flows into the cell. We learned in chapter 5 that calcium really is a messenger. And so it messages the presynaptic cell to do what? exocytose. It says, send your vesicles to the axon terminal membrane and exocytose. So calcium signals that at the axon terminal. So we exocytose. The neurotransmitter, are they all cute purple triangles? No, they're just there so we can visually see. Once the neurotransmitter, whichever it may be, epinephrine, norepinephrine, serotonin, dopamine, pick your neurotransmitter, it's going to be released into the what? Synaptic cleft, and hopefully bind with the receptor. This also takes us back to chapter 5 in the signal transduction. The receptor may be a gated channel. It may activate a G protein. It may, right, we can just keep going on for chapter 5 scenario. That depends on the postsynaptic cell. Is it a nerve cell? Is it a muscle cell? Whatever the case may be. I think it was this class that asked, well, then what happens to the neurotransmitter? Well, what happens is a few things. One, we can see that it may be what? What can happen to it? We can just break it down. Okay, well, we could just use an enzyme and break it down. What else is added? It may just diffuse on out of the cleft and be to an area where there are no receptors. And then say our astrocytes go clean it up, the microglia, think about it, clear it out. Additionally, our neurotransmitters may be, they're called reuptake transporters. So they may be taken back into the presynaptic cell and then reprocessed and repackaged, reused. These cells are green, reuse, recycle. And so we can recycle them, basically. We know that the cells, we identify this postsynaptic cell has to do something. So what's new here is the incorporation of the voltage-gated calcium channels. They have to be open with the activation of an action potential. Flux that calcium in. A whole series of reactions happen, but the net effect is exocytosis of those neurotransmitters. And then we can have an effect at the target. Let's just figure 8.2b. You have any questions before we look at some other images? I have a scanning image. We addressed what happened, right? Someone sliced the very tip of an axon terminal and exposed the vesicles, and they've been colored for us. Obviously, your cells like green and blue. But so this axon, presynaptic cell, axon has been colored green. We can see a bit of the membrane in the orange, internal portion. And the vesicles with the neurotransmitter are highlighted in the blue. So we can't see the neurotransmitter, but we can see the liquid vesicles. So they're in high concentration at the axon terminal. So we know that hydrophilic messengers can be stored, just like our neurotransmitter. This is what was not showing. All right, so if we kind of look at what's going on, we got some variants here. Reminding you, we're looking at nerve cells in the brain or the spinal cord. As in the previous image, the action potentials will be shown by flashes of yellow, but we know that's not literally what happens. But if your brain is trying to convey a message to, say, your spinal cord, for example, we can see how neurotransmitter can be released from the presynaptic cell. What color is the presynaptic cell in this image? The green, and the postsynaptic is purple. You can see the receptors as the little purple studs on the postsynaptic cell. We can also see the vesicles stored in the presynaptic cell. The voltage-gated calcium channels are highlighted in the green. So an action potential voltage pulls open those gated calcium channels. Calcium floods in, sets those vesicles into motion. Exocytosis, the neurotransmitter, which is shown here in the red things. They diffuse across the synapse, and they would bind with their respective receptor. Depending on that receptor structure is going to dictate whether or not that postsynaptic cell would either, say, hyperpolarize, letting potassium flow which way? Out of the cell. It can cause hyperpolarization. Or a different receptor may cause depolarization, so we would let sodium flow into the cell. Once that neurotransmitter,